Dr. D, 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 Dr. D. Explain stuff.
Hey everyone, Dr. D here. In this video we're covering Chapter 4, Microscopy, Staining, and Classification. So let's get started. The microscope allows us to see specimens that are smaller than what you can see with the naked eye. And I just want you to realize that the metric system is quite important when you're talking about microscopes because you need to understand just how small these organisms are. You all know what a meter is, right? It's about 3 feet. Well, a millimeter is 1,000th of a meter, isn't it? You know what a millimeter is. It's a tiny little space. A millimeter. So look here on your notes. A millimeter is 1,000th of a meter, 10 to the minus 3, 1,000th of a meter. Now, the naked eye can see particles that are about 0.2 millimeters big. Did that make sense? 0.2 of a millimeter. So you know what a millimeter is. Slice that into 5 parts, and that's what 0.2 of a millimeter is. Okay. Your eyeball has the resolution limit of about 0.2 millimeters. So if something is smaller than 0.2 millimeters, you can't see it with your naked eye. You can't resolve it with your eye. And you're going to need the assistance of a microscope. Did that make sense? Now, we're going to look at things that are smaller than millimeters in this class, right? What's smaller than a millimeter? Well, the next level down in the metric system is the micrometer. Micro. Here, the symbol for micro is mu, the Greek letter mu. It looks like a U, but with 2 tails, right? Now, the same difference between a meter and a millimeter, that means 1,000 times smaller. A micrometer is 1,000 times smaller again. It's 1,000 times smaller than a millimeter. So there's 1,000 micrometers in a millimeter, and there's 1,000 millimeters in a meter. Does that make sense? So you're dropping by 1,000 every time in size. That means 10 to the minus 6 meters. Okay, and most microorganisms are going to fall in the micrometer range, in the micrometer. So bacteria, archaea, those types of prokaryotes are usually 5 to 10 micrometers, whereas eukaryotic single-cell creatures, eukaryotes, are bigger. They're usually around 40, 50, 60, 70 micrometers. So we're really going to be worrying about micrometers. And viruses are going to be even smaller than that. Viruses are going to be in the nanometer range. Nanometers are 1,000 times smaller than micrometers. So that would be 10 to the minus 9 meters. Does that make sense? So the things that we are going to be studying in this class are mostly in the micrometer size range. And if we're talking about viruses, they're even smaller. They're in the nanometer size range. Does that make any sense? Hopefully it does. Let me know if it doesn't. Now, again, a micrometer. Now, let me tell you something else. If I say 1 millimeter, that's actually the same as saying 1,000 micrometers. Does that make sense? 1 millimeter is 1,000 micrometers. And so, remember what I said the resolution limit of your eyeball is? That's right, I said it's 0.2 millimeters, didn't I? 0.2 millimeters. How many micrometers would that be? 0.2 millimeters. Think about it. If 1 millimeter is 1,000 micrometers, what's 0.2 millimeters? 0.2 millimeters would be 200 micrometers. See what we set up here? The larger organisms, protists, are about 200 micrometers. And that's about the resolution limit of the eye. So your eyeball can just barely see some of the bigger single-cell eukaryotes, the protists. Okay, does that make sense? All right. So 200 micrometers is a large microorganism, you could say that. But we're going to need microscopes in order to see any smaller than that. Now, take a look here. We've been talking about this. You guys know how big a chicken egg is, right? It's in the millimeters range, right? It's in the millimeters range. And then fleas. Fleas, you know, with your eyeball, can you see a flea? Yes, but they're tiny. They're in the, I would say, 4 to 12 millimeter range, these fleas. So you know how small fleas are. They're barely able to be seen. So with the unaided human eye, which means 200 micrometers or more, you're able to see chicken eggs, you're able to see fleas, you're even able to see some of the bigger protists. Remember, these are protists, like the large Euglena protists, okay? But if we're talking about human red blood cells, if we're talking about chloroplasts of plant cells, if we're talking about mitochondria or bacteria, archaea, look, these are in the lower micrometer range. Can you see that with your eye? No, because remember, the unaided eye is only good down to the resolution of 200 micrometers. When you're dealing with red blood cells, we're talking about 9 or 10 micrometers in diameter. If we're talking about bacteria, we're talking about just about 1 micrometer to 3 micrometers in diameter, in size. So we're going to need a compound light microscope. By the way, the microscopes we use in a typical microbiology lab, these microscopes are called light microscopes or compound light microscopes because they have a series of magnifying lenses, right? Do you know what is the maximum magnification of the microscopes we use in the lab? Do you guys know? Do you remember what is the magnification of the ocular, the eyepiece? Remember, the eyepiece has a magnification of 10x, right? And what's the magnification of the objectives? Remember, there's a 4x scanning objective lens, there's a 10x low power objective lens, a 40x high power objective lens, and isn't there a 100x oil immersion lens as well? So what is the maximum magnification that you can achieve with a compound light microscope, the microscope that we are going to be using in the lab? Think about it. Well, our most powerful objective is the 100x objective, and our ocular, the eyepiece, is 10x, so the total magnification will be 1000x, right? Because 10x times 100x. So what you need to realize is the microscopes in our lab allow you to magnify the image up to 1,000 times, which means you can see objects 1,000 times smaller than you normally can. So if the resolution limit of your eyeball was about 0.2 millimeters or 200 micrometers, that means with the help of a light microscope you can see 0.2 micrometers. You can see all the way down here to 0.2 micrometers. Does that make sense? You can see 1,000 times smaller. So what are you going to be able to see with the light microscope? Down to 0.2 micrometers. That means you're going to be able to see bacteria and archaea. You're going to be able to see mitochondria, chloroplasts, human red blood cells, larger protists like the protozoan Euglena, right? That's what you're going to be able to see. It's perfect for looking at cells. What aren't you going to be able to see? Well, you know what's even smaller than 0.2 micrometers? Yeah, that's right. Viruses, ribosomes, proteins, individual proteins, DNA, RNA, things like that. That's smaller than 0.2 micrometers. That means that you're going to need fancier microscopes. You're going to need, and by fancier microscopes, I mean electron microscopes, right? Microscopes that are using, instead of light, they use electron radiation in order to resolve an image. So we'll talk about this, but just realize that with the light microscope, the compound light microscope, The smallest things you're really going to be able to see are bacteria and archaea. Anything smaller than that, you're really going to need more fancy optics. You're going to need electron microscopy for the most part. Does that make sense? All right, so let's talk a little bit about the general principles of microscopy and some of these terms that you need to understand to know how a microscope works. First of all, you need some sort of radiation, some sort of energy in order to resolve an image. With a light microscope, the radiation is called visible light. And remember, visible light are the energies between the wavelengths of about 350 nanometers, that would be violet light, to 700 nanometers, that would be red light. And we're going to cover that in the next slide. And with an electron microscope, the radiation would be electrons. So you have some kind of radiation that's based on wavelength. And you're going to use a microscope for what mainly? For magnification and resolution, right? That's what you want to, that's the usefulness of a microscope. A microscope magnifies the image, and the microscope allows you to gain better resolution. So what's the difference between magnification and resolution? They are definitely not the same thing. Magnification is how many times larger the specimen appears than it really is. If I have 2x magnification, that means the specimen is twice as big as it actually is. It appears twice as big. If I have 100x, you get the point. The specimen appears 100 times bigger than it actually is. What's resolution then? Resolution is the ability of a lens to separate and distinguish between small objects that are close together as separate and distinct. So what does that mean? Imagine you have two tiny dots, and those two tiny dots are getting closer and closer and closer together. At what point are you going to see one dot? Even though the two dots are not touching, at what point do you see one dot, right? That's the resolution limit, okay? So the closer the two tiny dots can get to one another, the better your resolution is. And remember what I told you the resolution limit of your eyeball is? That's right, 0.2 micrometers. If two tiny dots are closer than 0.2 micrometers, which means, oh, this is actually, of the light microscope. Yeah, this didn't look right to me. Let me show you something. I'm going to add here, and you could add it to your notes if you like. The resolution limit of your naked eye is what? Is, remember, 0.2 what? Millimeters, isn't it? Isn't it? Oops, let me swap these. The resolution limit of your eyeball is 0.2 millimeters. You know what that means? If two small dots are closer than 0.2 millimeters, your eyeball is not going to be able to distinguish that they're actually two separate dots. Your eyeball will see one dot. Does that make sense? But what did I tell you you gained with the microscope, with the compound light microscope? Remember I told you with the compound light microscope, you can do 1,000 times magnification and resolution. So with the help of a microscope, now what's your limit? What's your resolution limit? That's right, it's 1,000 times better than 0.2 millimeters, which means now with the help of your microscope, you can go to 0.2 micrometers. Does that make sense? Does that make sense? Because 0.2 micrometers is 1,000 times smaller than 0.2 millimeters. So that's why we use, if everyone, like, this is why we use microscopes in the lab, because now instead of trying to squint really hard and look at a little tiny particle and see tiny little things with our eyeballs, which aren't all that good for looking at microorganisms because we can only resolve down to 0.2 millimeters. Now with the help of this compound light microscope, when you set it to 1,000 times magnification, now your resolution is down to 0.2 micrometers, 1,000 times better. So now I can see bacteria. Now I can see archaea. Now I can see red blood cells. Does that make sense? Ooh, here's a quiz too. Quick test your knowledge quiz. 0.2 micrometers. What's another way of saying 0.2 micrometers? Do you remember 0.2 millimeters? Let's test you on this one. What was another way of saying 0.2 millimeters? That's right, 200 micrometers, right? Check back in the notes. So what's another way of saying 0.2 micrometers? That's right, 0.2 micrometers is 200 nanometers, okay? These are just things that you should be aware of, that you should understand. Okay, this is good stuff for you to know, especially if you're going to be a nurse, especially if you're going to be a nurse anesthetist, definitely. If you're going to be any kind of pharmacist tech, you're going to need to know these values, micrometers, micrograms, okay, things like that. Very, very important in medicine. So anyway, now you know how the metric system works a little bit, or at least it was a good refresher for you. So what's contrast then? We can't lose sight of contrast. Contrast is the difference in intensity between two objects. So if I have a specimen against the background, how well does the specimen stand out from the background? If the specimen blurs into the background, so I can't really tell what's background and what's specimen, that means I have poor contrast. Does that make sense? But if the specimen is very distinct from its background, if the specimen really stands out, like let's say the specimen is bright red and the background is white, that's good contrast. Does that make sense? So if a sample has bad contrast, what can we do in the lab? I hope you said staining, right? See, you know how we stain cells? You can stain cells purple, you can stain cells red, you can stain cells green, you can stain cells blue, various colors, right? Why do we stain cells all these colors? Well, because without staining, a lot of times the cells don't stand out against the background, and we have poor contrast. Now, look at this. Remember back here, I just want to show you something. Remember back here when we're talking about general principles of microscopy, I was telling you about wavelength of radiation, and I was telling you that microscopes use wavelengths of radiation, of electromagnetic radiation, energy, if you will. We need to understand how wavelength of radiation works. So here's the electromagnetic spectrum, right? This electromagnetism works according to wavelength, right? And energy. Some waves are as small as 10 to the minus 12 meters, okay? 10 to the minus 12 meters. You know what that means? 10 to the minus 12 meters? That's even smaller than a nanometer, okay? 10 to the minus 12 would be even smaller than a nanometer. It's 1,000 times smaller than a nanometer. That would be, I believe, a femtometer, okay? A femtometer. That's tiny, okay? Or is it picometer? It's either picometer or femtometer. It's tiny, man. So look at this. 10 to the minus 12 meters is a very, very short wavelength. Those are gamma rays, okay? Then we have 1,000 times bigger wavelengths. These are your X-rays, which are still very small waves. Then you have UV waves, which are bigger than that. And then here in the middle, you have what's known as the visible light spectrum of electromagnetic radiation. Visible light, the light you see, all the colors that you see with your eyeballs, that's visible light. And that falls under the wavelengths of 400 nanometers, which means violet or indigo, all the way to 700 nanometers, which is red, okay? And the difference in color is simply a different wavelength along the spectrum, okay? So blue light is here. Green light would be here. Green light would be halfway between 400 and 700 nanometers. Yellow light is a little further, a little bit larger than green wavelength. So this is how you define colors of light, right? And the light microscope, by the way, the compound light microscope, uses visible light in order to resolve the image. Okay, but an electron an electron microscope uses electrons to resolve the image, and electrons are far smaller than the wavelength of visible light, and that's why you can get more magnification with better resolution. Does that make sense? Hopefully it'll make sense later. Then you have infrared. Infrared is longer wavelength than visible light. Microwaves are an even longer wavelength, and radio and television waves are meters big in wavelength. So this is the spectrum. Here's what I want you to know about the spectrum. This is what I want you to know. The shorter the wavelength, the better the resolving power of the microscope. So if a microscope uses gamma rays, I don't know any gamma ray microscopes, but if a microscope were to use gamma rays, it's going to have a lot better resolution than if you use radio waves or television waves. Does that make sense? So the shorter the wavelength of the electromagnetic radiation you use for your microscopy, the better the resolution will be for your microscopy. And another thing I want you to know is shorter wavelength energies have higher energy, right? So gamma rays and X-rays and UV light has higher energy than radio waves, television waves, microwaves. Does that make sense? What else do I want you to know? Those are the two main things that I need you to know. And I need you to know that your compound light microscope uses visible light between 400 and 700 nanometers, whereas electron microscopes use electrons, which are smaller, much smaller in wavelength. And we're going to talk about how small those electron waves are. So if we look at a compound light microscope, you'll notice that it has lenses. It'll have these convex lenses. And what does convex lens mean? It means a lens that bows out, right? It bows out. Convex lenses bow out. Concave lenses would bow inwards, okay? Convex lenses allow for magnification because when the light from this is the specimen or sample, light from the specimen reaches the convex lens, and then through a process known as refraction. Refraction is bending of light due to slowing of light. When light goes from one refractive index, air, to another refractive index, glass, which has a higher refractive index. You don't need to know this for the purposes of Biology 2420, but for Biology 2421, which would be for science majors, you may want to understand that refractive index matters. So air has a refractive index of, let's say, 1. Glass has a refractive index of 1.5. So because of that, when light strikes glass, because it's going from one refractive index to another, that causes this bending of the light because there's a bend towards what's known as the normal. Again, you don't need to know this from my class, but what you do need to know is that light bends when it strikes glass at an angle, and it bends towards this normal. And when light bends, it magnifies the image, right? It magnifies the image, and the image is going to be inverted and reversed and enlarged to your eyeball. In fact, let me show you something. We're going to come back, but look at this. Again, when a ray of light passes from one medium to another, refraction occurs. That means light is bent at the interface, and remember, it's bent towards the normal. What's the normal mean? The normal means perpendicular to the surface of the plane. You don't need to know that from my class, but the refractive index is a measure of how greatly a substance slows the velocity of light. So again, When light goes from lower refractive index, which is air, it strikes glass. Glass would have a higher refractive index. This would cause the bend. This would cause the bend. And that's why the light bends, right? And so if you have like a piece of glass, let's say like a window, when the light hits the glass, because it's hitting right at the normal, which means the light's hitting right at perpendicular, the light doesn't bend, because the light has to hit at an angle, right? So the light hits here, this convex lens, convex, bowed out. When the light hits a convex lens, it will bend. Why? Because it bends toward the normal. And when it does that, it comes to a point, and that's called the focal point. Okay? If you have a thicker lens, a more rounded convex lens, that means that the light gets bent even more drastically. So your focal point is even closer. If you've ever noticed with the microscope, let me tell you something. With the microscope, if you're on 4x objective, the objective lens is quite far from your specimen, right? But when you go to 10x objective, when you switch it to the 10x objective, did that objective get closer to the specimen or further away? It got closer, right? Why is that? Well, because the 10x lens is thicker, so it's going to have a closer focal point, and it's going to be closer to the specimen. Now when you switch it to the 40x objective, that lens is even closer to your specimen. And when you switch it to the 100x objective, well, that's when you need oil. Why? Because that lens is basically touching the specimen, right? Your oil lens is so thick that it's basically on top of the specimen. It's almost touching the specimen. And in fact, that's when you have to add the oil. So what happens if you have a concave lens? Remember, bowed inwards? Concave lens, the opposite. When the refraction occurs, the light scatters, right? So concave lenses would not be good for microscopes, right? Because you wouldn't want to scatter the light. You want to bring it to a focal point, right? The specimen would be here. Your eyeball would be there. You'd see the image. So again, here is your oil lens, right? Here's your oil lens. Here's your specimen. Light comes in through the specimen into the lens, and then you see the specimen under the microscope. But do you remember with the oil microscope, don't we add some immersion oil to that? Why do you think we add immersion oil for the oil lens? Well, remember what I told you. The oil lens is quite thick. It's a convex lens that's very thick. That lens is very close to the specimen. So it's a very thick lens, very close to the specimen. When the light hits the specimen, it will bend. And some of that light will bend away from the thick objective lens. So that light escapes. But if you put the oil there, the light doesn't bend away. It goes straight into the objective lens. Why do you think that is? Well, because immersion oil has the same refractive index as glass. Does that make sense? So there's no reason for the light to bend if it's going from glass, which has a refractive index of, let's say, 1.5, into immersion oil, which has the same refractive index. Light will travel straight. Light will never bend. Remember, light only bends when light's traveling from a medium with one refractive index, let's say air, which has a lower refractive index than glass. Well, when light goes from air into glass, it's going to bend if it hits at an angle, right? But if light is going through oil and it hits glass, or from glass and it hits oil, because they have the same refractive index, even striking at angles, there's no bending occurring, okay? I know this is kind of hard to grasp, but so let me know in the comments below if you have any questions. I'll be glad to answer them. But again, for Biology 2420, I don't need you to know exactly how the light bends, but I do need you to understand the concept of refraction. You know, how come when you put a straw, you know, like a drinking straw, into a glass, how come it looks like the straw is broken? How come it looks like two straws? You know, or How come when you put a stick into the swimming pool, how come it looks like the stick's broken and kind of going off at an angle? Like what? Why? Well, that's called refraction, right? That's all due to refraction. And refraction is very important if you're going to understand how lenses work. I just need you to know that because of refraction, lenses can bend light. And because they can bend light, you got these focal points and you can magnify images. And the thicker the lens, the more magnification you get, and the shorter the focal point. That's what I want you to know. Thicker convex lens, shorter focal point, more magnification. Okay? Thinner convex lens, less magnification, longer focal distance. Does that make sense? Good. Now, let's go back to this slide here. So at this point, I wanted to talk about the types of microscopes that are out there, starting with the simple microscope, the simple light microscope. This is a type of microscope that only has one magnifying lens. If you think back, do you guys remember I told you about Leeuwenhoek, Anthony van Leeuwenhoek, and his simple microscope, his pinhole microscope? That microscope only had one lens and it magnified an image. So this is about as simple as microscopes get. However, remember that in our lab and in most microbiology labs, we're dealing with a compound light microscope, meaning that there are two sets of lenses. Remember, and by two sets of lenses, two sets of magnifying lenses, right? One set are called the objective lenses, and usually you have a 4x, a 10x, a 40x, and a 100x compound lens. And you can switch between them, right? Because they're on this rotating nose piece here. You see you have the 4x, you have the 10x, 40x, and 100x. And you guys remember what I was saying earlier? The 4x is a really short, stubby objective, right? When the 4x is in place, it's quite far from the specimen. Remember why? Because it's got the thinnest lens. It's got a thin convex lens. Whereas when the oil lens is in place, remember the oil lens is almost touching your specimen. It's a really long objective. Why? Because it has the thickest lens, and that lens magnifies a bunch, but it also needs to be right on top of the specimen because it's got the shortest focal length, right? Anyway, this is called a set of lenses. The objectives are one set. What's the other magnifying lens on the microscope, on the compound microscope? That's right, the ocular lens. These oculars magnify as well, and usually you have 10x magnification. Oculars magnify by 10, objectives magnify by whatever it says on the objective, 4x, 10x, 40x, and 100x. And how do you calculate total magnification? Well, on the 4x objective, if the 4x is engaged, it would be 4 times 10, right? So 40x is the lowest magnification possible on this microscope. And what's the highest magnification possible on this microscope? That's right. If you have the oil lens engaged, the oil lens is 100x times 10 for the ocular. So 1000x is the highest magnification with this microscope, and 40x would be the lowest magnification with this microscope. One more cool thing about the compound light microscope is that once you get a specimen in focus with one lens, when you switch to the next lens, it's still almost in focus. The term for that is called parfocal. Parfocal means a specimen remains in focus or nearly in focus as you change objective lenses. As long as you're in focus with one lens, you should be almost in focus with the other lenses. And good compound microscopes these days, most of them are calibrated so that they are parfocal. So again, compound light microscopes are used in the lab. They have two sets of magnifying lenses. But there are different kinds of compound light microscopes. There's the bright field, the dark field, the phase contrast, fluorescence, and confocal. So you have different flavors of microscopy, of compound light microscopy. So we're going to talk about each one. What is bright field? What is dark field? What is phase contrast? In fact, let me tell you something. The microscopes in our lab are actually capable of bright field microscopy, dark field microscopy, and phase contrast microscopy. How do we go from bright field to dark field to phase contrast? Well, look at this. Do you see this piece here that I'm outlining? This piece right here? That's called the condenser, right? The condenser, which focuses light through the specimen. This condenser has various lenses in it that you can switch around. This condenser can switch between bright field, dark field, and phase contrast. Does that make sense? So you can switch this microscope in the lab from dark field to bright field to phase contrast, so you can experience 3 different types of microscopy with this microscope right here. However, fluorescence and confocal microscopy, these are a little bit fancier, and you're going to need some, more powerful optics, lasers, I should say, in order to use these. And I'll talk about these later, but you should know that our microscopes are capable of the first 3 types of microscopy. So what is bright field microscopy? What is dark field? What is phase contrast? Let's talk about that next. So again, with bright field, this is a setting on our microscopes. This is done with compound light microscopes. You have a series of lenses for magnification. Remember, you have objective lenses and ocular lens. Light passes through specimen, then through the objective. And oil immersion has the highest. And this is really the most simple type of compound light microscope, the bright field microscope. You know why? Light travels through the specimen. It travels through the the objective lens, the ocular lens, and then you directly see light through the specimen. There's direct light through the specimen. So look at this picture at the top here. This would be like bright field microscopy. The light goes through the condenser, the light goes directly through the specimen, and the light is not manipulated in any way. The light traveled through the specimen, up through the objective lens, up through the ocular lens to your eyeball. Very simple and straightforward. Nothing really to it. However, bright field is not ideal for viewing unpigmented and unstained cells, right? Look at this cell here. Look at these. The reason you can see these diatoms, these diatoms, these green algae, the reason you can see these is because these are pigmented green because of their chloroplasts. So that's why it's so easy to see these structures. And what's the term for that? Easy to see a specimen against the background. Remember, what was the term for that? That's right, it's contrast, right? You have nice contrast because these cells are naturally pigmented. These cells are naturally green because of the chloroplast within. But what if they didn't have chloroplast? What if you were looking at some cell that's not colored at all? Then you wouldn't have good contrast, right? So what you need to understand is bright field's great and all, but not ideal for viewing unpigmented, uncolored, unstained cells. So what if you want to see living cells but they're unstained? Well, then these types of microscopes are more useful. Dark field, which again our microscopes are capable of switching to by switching a setting on the condenser. Phase contrast, which again our microscopes are capable of switching to. And DIC, or differential interference microscopy. Our microscopes in the lab aren't capable of DIC. However, that's another trick that you can play in order to gain more contrast for your microscope. Okay, so let's talk about how these types of microscopy work and why they increase contrast. Okay, so again, what's the limitation of bright field microscopes? They aren't ideal for viewing unpigmented cells, especially living unpigmented cells. That's why you would want to switch to a dark field. That's why you would want to switch to a phase contrast or a DIC. So let's talk about that. So what is a dark field microscope? Let's say, again, I'm looking at some cells under the microscope that are alive, but I can't see the specimen too well. I might want to switch to dark field by sliding that condenser setting, sliding that condenser over to dark field, and I'm going to look at these pale objects. Now the pale objects stand out really nicely against the background because the background appears dark. The background appears black, and the specimen appears lit, like bright, usually a white color. So now you have much more contrast. And how does this work? Why does this work? I wonder if I have an image here. I may not, but I want you to, what I want you to know is the way dark field microscopy works is that when you switch it to this type of condenser setting, the condenser makes all of the light escape the objective. Let me show you. Look, let me show you. You see how this is, you see how the light is going through the specimen into the objective lens? That would be bright field microscopy. Does that make sense? Now, if I switched it to dark field microscopy, the light would hit the specimen, and then the light would just leave. The light would not get captured into the microscope. Does that make sense? So the light coming in would just leave, and so you would see black. But guess what? If light were to hit the actual specimen that you want to study, the light would get refracted or bent inwards so that it would be captured by the objective lens and viewed under the specimen. So you know what you would see? You would see something like this. This is a pale cell. This is a pale cell with bright field. You see how this bright field cell is so pale and their contrast is really bad? You can't really see the cell. If I were to switch it to dark field, look what I see. I see nice contours now. I see a nicely lit cell against a black background. And why did I get that? Well, because all of the light escaped the microscope, escaped the objective, except for the light that struck the specimen, which was refracted into the objective lens. Okay, does that make sense? You see how much better the contrast here is with dark field? So what do I want you to know about dark field? I don't need you to know the ins and outs of exactly how dark field microscopes work, but what I do need you to know is that dark field is a condenser setting on a compound light microscope that increases contrast by providing a black background or dark background and a lightly colored specimen. Okay, because only the light that hits the specimen reaches your eye. Does that make sense? Hopefully that does. Again, if it doesn't, let me know in the comments below. I'll try to explain it in a different way. Okay. Now, how does phase contrast microscopy work, or phase microscopes? Very similar way. In that the condenser, let me show you, the condenser changes how the light works. So in this case, the condenser changes the phase of the light that hits the specimen. We don't need to know exactly how this works for this class, but let me explain it just in case you're curious. Light travels in waves, and the waves are parallel, so they're traveling together. Okay, if light is in phase, the peaks and the peaks and valleys are, uniform. But if light is out of phase, that means some waves are going in alternating waves, right? So when one light wave is cresting, the other one is going down the valley, and so the light is out of phase. And when light is out of phase, light can get canceled, right? It appears as a dark spot. Okay, now you don't need to know this for the class again, but what you should realize is, with the help of phase contrast microscopy, an stained or unpigmented cell has more contrast. Why? Because the phase contrast microscope made the image appear darker than it really is. How? By changing the phase of light and causing that light to cancel, right? So light that hit the specimen, this light here, is going to get out of phase. And when that light gets out of phase, that light appears darker, right? That specimen appears darker. So phase contrast microscopy is an artificial way of staining a cell. You're not really staining a cell, but you're using light tricks to give you the impression of a stained cell. Does that make sense? So this would be bright field, the light's going through the specimen, and you have really poor contrast. This is phase contrast. The light that's hitting the specimen is forced to become out of phase, and therefore you see a dark image, a dark specimen against a lighter background. Okay, very interesting stuff. Now, what's a DIC microscope? Differential interference contrast microscope. Again, you don't need to know exactly how this works, but let me explain it just in case you're curious. Have you ever gone to the movie theater and you want to watch a 3D movie? And you know how 3D movies work, right? Nowadays, they give you these special glasses. And I'm not talking about the old 80s glasses, which have the blue lens and the red lens. We're not talking about that. How about glasses that are polarized? They're gray glasses, you know, gray lenses. You put on these glasses, you sit back and you watch your movie, and the you know, when someone throws something, it looks like it's coming right at you, and you're like dodging stuff, and you're getting all excited at the movies because everything's 3D, right? How does that work? Well, the way that works is due to the way that light is polarized, right? You've got polarized light. Some light travels in this axis, the y-axis, and some light travels in this axis, the x-axis, right? So if you can change one eye to only receive, you know, y-axis light, and the other eye only receives x-axis light by having polarized lenses, okay, you can see 3D images, can't you? So with this type of microscope, you're going to see 3D images. You're going to see like a 3D amoeba. Why? Because it uses polarizers similar to what a movie theater does. It allows this eye to get one information, that eye to get the other information. Suddenly you're seeing a 3D image. And of course, anytime you can see a 3D image, you're going to get a better sense of contrast than if you don't, right? So interesting stuff. Again, you don't need to know exactly how that microscope works, but what you should realize is with the differential interference contrast microscope, you're going to get some better resolution. Better contrast. And the reason you get that nice looking image, that three-dimensional looking image, is because of polarity of light, right? There's a manipulation of the polarity of the light by the microscope. It's really interesting stuff. So look at this. This is the same image. This is the same image with bright field. Notice that really poor contrast. With dark field, notice the black background and light image. With phase contrast, notice that the sample is dark. And with differential interference contrast, or DIC, notice that the image looks three-dimensional, looks almost like a topographical illustration. You can see the topology, you can see the surface of the structure, and see all the little peaks and crests and little valleys, and you know, all the parts of the cell and how they kind of are shaped. Interesting stuff. So all of these are types of compound light microscope. Okay, now let's talk about fluorescence microscopes, which are another type of compound light microscope. However, we don't use these in the lab, mainly because, you know, they're kind of expensive and they require a lot more prep work to prep your sample. I'll explain why in a second. But let's take a look at what these fluorescent microscopes are. So with fluorescent microscopes, you're going to need a powerful light source, usually a UV light source, or some kind of laser. And that, these can be expensive light sources, but the reason you need these powerful light sources is because um, these these lasers allow for the fluorescence to occur. Fluorescence is when a molecule absorbs light at one wavelength, becomes excited, and then emits light at a different wavelength, usually, or actually always, a longer wavelength. So um, let me explain it this way. Let's say there's a pigment. Okay? If a pigment is a fluorescent pigment, if I were to shine a particular wavelength of light at it, let's say blue light, I shine blue light at this pigment can become excited by the blue light, and when it becomes excited by the blue light, it might emit its own light, and that light is a longer wavelength light. For example, a green, a yellow, an orange light, or a red light. Does that make sense? I know it's kind of tricky, but you don't need to know exactly how fluorescence works. I'll tell you what you do need to know. So look at this specimen here. If I were to look at this specimen with fluorescent microscopy, I would see glowing parts, right? This is where the fluorescence is. I'd shine blue light at this specimen, but my specimen would get excited and emit green light. Okay, and why did it emit green light? Well, usually because I added something to my specimen which is fluorescent. Like I added a molecule that is fluorescent, but it binds to the DNA of the cells. Okay, these are called fluorophore molecules. These are called fluorophores. So for example, let me show you something. If I were to look at bacteria, if I were to look at this bacteria under the microscope, under a fluorescent microscope, I would see nothing. Does that make sense? Because bacteria don't normally fluoresce. Bacteria don't naturally fluoresce. What does that mean? I could shine lasers at bacteria all day. They're not going to start glowing. Does that make sense? However, if I were to be able to take a fluorescent dye or a fluorescent, they call them fluorophore, these molecules that are fluorescent, take that fluorescent dye, attach it to an antibody, then have the antibody attached to the bacteria, well, suddenly, what do I have? I have a bacteria that's covered with antibody, which is conjugated to or linked to dye. Does that make sense? Now what happens when I shine the blue light onto my specimen? My specimen glows, it fluoresces, right? Does that make sense? So if I want to see these bacteria, what do I do with fluorescent microscopy? I want to label it with something that glows. I take the fluorescent dye, conjugate to antibody, antibody conjugates to bacteria. Now I can see my bacteria. So that's a lot of work, right? You can see why we don't do this in the lab, can't you? You can see why we wouldn't want to do fluorescent microscopy in a very simple BIOL2420 lab. Well, because we've got to prep our samples, we've got to find the right antibodies for what we're staining, we got to find the right fluorophores, we got to have access to these expensive lasers, these UV lights and whatnot. Okay, so again, do I expect you to know everything about fluorescent microscopy? No, it's quite complicated. But what do I want you to know? What do I need you to know? Well, what you should know is that fluorescence microscopy allows you to see parts of a cell. Okay, why? Because of these fluorophores. Here's the word fluorophores. And fluorophores are always excited at one wavelength and emit light at a different wavelength. Do you remember what I said? If I shine blue light on a fluorophore, it might get excited and emit a different wavelength. And remember, is that wavelength higher wavelength or a shorter wavelength. Is it a longer wavelength or a shorter? It's longer. The emission wavelength is always a longer wavelength than the excitation wavelength. So what does that mean? Again, if I want fluorescence, I'm going to shine a shorter wavelength light, and the specimen is going to give off a longer wavelength light every time. Does that make sense? So that's all I want you to know about that. I want you to know what a fluorophore is. Here I define it. A fluorophore is a molecule capable of fluorescing. Without these fluorophores, you're not going to see anything with a fluorescent microscope. And fluorophores always get excited at a shorter wavelength than they emit. That's what I want you to know. Now let's talk about confocal microscopes. This is a type of fluorescent microscope, so you need fluorophores for it. However, what do I want you to know about these compound light microscopes? These types of compound light microscopes, the confocal microscope. I need you to know that you can make a 3D image from digitized images. Let me show you. With confocal microscopy, you're going to be able to make a 3D image of, your specimen. So it's basically the best of both worlds. You've got fluorescent microscopy, but you can also have a 3D version. So here on this slide, you can actually see some of my work from graduate school. When I was working on my PhD, I was trying to understand how the pancreas works and how insulin is produced in the pancreas and how that might lead to diabetes. So I had different mice. You don't need to know this for the exam, but I just thought you might find this interesting. This was some of my own research. So that's my hand right there. I'm holding 3 mice, a wild type, which means a normal mouse, and then a mutant mouse. And the mutation was a change in a gene called BMP receptor 1A. Obviously, you don't need to know any of this stuff, but obviously this mouse is smaller. These were siblings, right? These were all from the same litter. This mouse is smaller. The normal mouse is bigger. So what's wrong with this mouse? We looked at the pancreas. Here's a normal pancreas. We looked at it with bright field microscopy, right? And then I looked at the pancreas with fluorescent microscopy. Look at that. There's the pancreas of a normal mouse, of this mouse on the left. The mouse on the right, notice how small the pancreas is, right? Notice how small the pancreas is. So the pancreas is much, much smaller. Maybe that's one of the reasons why the mouse can't grow as much, because you got less insulin. And not to mention, look at the stomach. The stomach here is much smaller as well. Here's bright field image of the stomach. And, I didn't actually label the stomach too well with fluorescent microscopy. So remember, with fluorescent microscopy, you can only see what you've labeled with those fluorophores. I labeled the pancreas with fluorophore. Okay, and then look at the sections of the pancreas. I sectioned the pancreas, and look here. A normal pancreas, this is called the pancreatic bud. A normal pancreas has a big bud here, right? The mutant, the bud failed to develop right. Does that make sense? This is during embryogenesis. This is when there's an embryo. And then I used all kinds of phase contrast microscopy to see parts of the pancreas. And then I used confocal microscopy to look at the blood vessels in the pancreas, you see? So this is just showing you that all different kinds of microscopy are used today. And I used all of these ones I've been talking to you about today. I've used all of them when I was working on my PhD. So it's not this abstract thing that you might never see. A lot of nurses do research, and they're working with microscopes, so you might be able to see a fluorescent microscope one day, or a confocal microscope one day, or a bright field microscope. Obviously in the lab we're going to see bright field microscopes. Oh, and by the way, look at this. In these pancreata, by the way, the plural for pancreas is pancreata, believe it or not. Not pancreases. Pancreases is not the correct term. It's pancreata. Yeah? You learn something new every day. When I looked at these pancreata, I saw that, I could see hormones in those pancreata. Like, are the pancreata producing glucagon, or are they producing insulin? the normal pancreas was producing insulin, and also my mutant pancreas was also producing insulin. So unfortunately for me, insulin was not the answer. Insulin was not disrupted in my particular mutation. So diabetes wasn't the reason why this mouse was so small. For me, the reason why was more this reason. A normal mouse forms a lot of vasculature, blood vessels, in the pancreas during development, whereas my mutant mouse was not forming the proper blood vessels during development. And that's why if you don't form blood vessels correctly, your organs don't develop as well or as large, and so you're going to be stunted in that way. So it was an interesting project, and I had to use all kinds of microscopy, the ones that we've learned about today, in order to study. But remember, I told you, if you want to look at stuff that's smaller than a bacteria, or if you want to see bacteria in really, really high detail, then you're going to turn to electron microscopy. You know why? Because with an electron microscopy, you're not just magnifying an image times 1,000, you're magnifying an image by 10,000 to 100,000. So think about it. An electron microscope allows you to magnify 100,000 times, right? 100,000 times, not just 1,000 times. Because of this, you're going to see detailed bacteria, viruses, internal structures of cells, molecules, even atoms. You can see individual atoms, right? So it's really, really interesting stuff. And there's two main types we're going to learn about: transmission electron microscopes and scanning electron microscopes. Let's talk about the difference between the two. Let's start it with a transmission electron microscope, TEM for short. What is a transmission electron microscope? This is a picture of one. You see how big they are? This here is a vacuum chamber where you suck out all the air. You cannot have air in a electron microscope. You know why? Your radiation source is electrons. You're shooting electrons at your specimen. Electrons don't travel in air, right? They don't travel well in air. Imagine if there's air floating around, air molecules, which are nitrogen, oxygen, and a bunch of other CO2 and other molecules. When you shoot electrons at air, the air absorbs the electrons. So you've got to suck all the air out, so you have a vacuum. And once you have a vacuum, the electrons can travel through the vacuum. So look, at the top here you would have an electron gun. Seriously, it's called an electron gun. And then it's going to shoot electrons. And those electrons travel from one side to the other, right, to this fluorescent screen. The screen will become excited by the electrons, right? The electrons hit the screen fluoresces, and that sends the image to a computer, you see? So the gun shoots the electrons travel to a screen, the screen translates the image onto a computer screen for you. Now, your specimen is right here. Your specimen, let's say here's a bacteria cell. Your bacteria cell is right here. So how do you prepare for this? First of all, you got to get your specimen prepared, right? So what do you have to do? You have to take your specimen, you have to dehydrate it so all the water is gone. And then you got to stain your cell. How do you stain a cell for electron microscopy? Well, you can't stain it with colors, right? Why would you stain it purple or yellow or pink? You're not going to be able to see colors, with an electron microscope, because it's outside of the visible spectrum, right, of light. So the way you stain for electron microscopy is with heavy metals. So you're going to use gold or silver or some kind of heavy metal. And look at this. The specimen is stained with heavy metal. So anywhere that the heavy metal coats, and by heavy metal I mean like a fine mist of individual atoms of metal, this metal attaches to the cell. So when the electrons travel through the specimen, if an electron gets blocked, you're going to see it as a dark spot on the specimen. Wherever you see light spots, that means there wasn't much staining with the gold or with the silver or whatever element you used. Wherever you see dark spots, that means that the electrons were blocked. Wherever you see light spots, the electrons went through. And by the way, with transmission electron microscopy, you have to section first. Again, you dehydrate, you stain with heavy metal, and you have to section the samples, right? Section the samples. So you can't see like a whole bacteria, but you could slice the bacteria into really, really thin, micrometer sections, right? Really tiny sections. So you section your sample, and then you stain with heavy metal, and then what you can see are internal parts of the cell. You see, this is probably the nucleoid. Here's a nucleoid, and then you can see all the different ribosomes. You can see all the different parts of this bacteria cell. So this is a bacteria cell that's dividing, right? You can see all the insides, the innards of this bacteria. Why? Because with TEM, with transmission electron microscopy, you've sectioned the specimen, and you've stained it with heavy metal, and you're looking inside, right? And you're going to get really, really high resolution with this microscope. By the way, do you think there are lenses, glass lenses inside of this microscope? How do you resolve the image? How do you control the electrons? How do you magnify? How do you control the electrons? Well, you can't use glass lenses. Like here on the left, you have a standard light microscope. Remember, a light microscope uses a light bulb, and then it uses glass lenses, okay, to magnify the image. You can't use glass lenses with a electron microscope. Why? Because a glass lens would actually block electrons. There's no way to use glass lenses. So instead, you use magnets, right? The lenses are actually magnetic lenses, not glass lenses. So magnets force the electrons to, magnify the image. Magnets force the electrons through the specimen and then cause the electrons to come to focal points and to magnify the image, right? Does that make sense? You're magnifying the image using lenses that are actually magnets. If you were to use glass lenses, no way. Because if light if light can travel through glass, electrons cannot. Remember, electrons can't even travel through air. So how are electrons going to travel through glass lenses? Doesn't work that way. So interesting fact for you. And here are, actually these are SEM images. So again, what's TEM? TEM is transmission electron microscopy. What kinds of things are you going to see? You're going to see the insides of cells, right? Because you had to section your cells. But what about SEM? Remember I said there's two types of electron microscopy you need to know about: transmission electron microscopes and scanning electron microscopes. Well, scanning electron microscopes, you're looking at topography. Look at this. You're looking at the surface of the cell. Does that make sense? The only difference between TEM and SEM is in TEM, you had to section your sample and then look inside. With SEM, you don't section the sample. Instead, you see the surface of the sample. Why? Because let me show you this. You remember your electron gun? Well, it would scan electrons across the surface of the specimen. And when you scan, look, if I were to scan across the surface of this specimen, I would see what the surface looks like. If I were to scan across the surface of this specimen, I would see what the surface of this specimen looks like. So what are SEM microscopes good for? What is scanning electron microscope good for? That's right, looking at surface structure, looking at topography. What are TEM microscopes good for? Looking for internal structures, okay? Now you might be wondering, but Dr. D, why are these colored? Like, I thought you said electron microscopes don't have color. Why Why are these streptococcus colored orange? Why is this guy this, what is this? Arachnoidiscus. Arachnoidiscus. This is a very strange, discus-looking creature. Now, what is this guy? And this Aspergillus, which is a type of mold. Paramecium, which is a type of protist. This is a type of protozoa. So, and look at the cilia on this paramecium. That's really cool. Look at the cilia, the short beating hairs on the surface of this creature here. Look how detailed. So why are all these colored, you might be asking? Like, how come all of these have different colors? Those are called artificial stained colors, artificial colors. With a electron microscope, your image is always going to be black and white, on a grayscale. And any color you see, like images like this from your textbook, that's an artificial, like basically photoshopped, right? It's something that you could add later, just so you know. And the last type of microscopy I really want to mention is probe microscopy. You don't even really need to know about this much. I just threw it in here just so you know. These types of microscopes go even more detailed. Like I'm talking 100 million times. I'm sorry, 100 million times magnification. So you're going to see individual atoms. You can even see like a hydrogen atom with this type of microscopy. There are two types: scanning tunneling microscopes and atomic force microscopes. So these are even more sensitive types of microscopy out there that are even more sensitive than electron microscopes, where you can see down to the atomic level, literally atoms. Like you could see a helium atom or H2. You can see hydrogen gas, right? You can see these types of things. These very, 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 very sophisticated and specialized microscopes out there. So you see, so you're going to see DNA. This is the scanning tunneling microscope image of DNA, for instance, or the atomic force microscope image of DNA and enzymes, right? So you can see all this stuff at very super detail. And there's a big chart here detailing some of the types of microscopy that we looked at and some of the important things you need to know about these types. I suggest just looking at these for an overview of microscopy. All right, so that wraps it up for part 1 of 2 for this chapter. Tune in to the next video where I'll be talking about staining. Thanks for joining me again. Hope you learned a lot. If you have any questions, throw them in the comment box below and I'll be glad to answer them. And I'll catch you guys next time.
Dr. D, Dr. D, Dr. D, Dr. D, Dr. D, Dr. D, a Dr. D, Dr. D, Dr. D, Dr. D, Dr. D, Dr. D, a Dr. D, Dr. D, Dr. D, Dr. D, Dr. D, Dr. D, a Dr. D, Dr. D, Dr. D, Dr. D, Dr. D, Dr. D.